Okay, so welcome again, my people. This is a part 2 of model 4. Yes, model 4. It's been a while. So in this video, we are going to look at different methods for ascertaining achromatic, whether a particular region of a genome is achromatic or heterochromatic. Okay, so we are actually going to base it on these 4 techniques here. Okay, so let's just look at like an overview before I take them one by one. So the first one over here, so we have DNA binding protein ChIP-seq, right? Histone modification ChIP-seq, DNA-seq, and FAIR-seq. I call it FAIR. Right. So the DNA binding protein is where probably you have a region of interest, okay, but your gene of interest is within that region, and you don't know where that gene is particularly. So you just use a protein that is actually that can bind to the gene of interest you are looking for, right? So that protein goes in, bind to wherever we need, then we subject the entire DNA to fragmentation, okay, so probably through sonication. Then those unbound by the protein will be fragmented, but not those with the protein. So afterwards you can release the proteins and get your gene of interest. Then you can go on to do further sequencing you want to do. Similar thing happens with the histone modification, but this one we are not targeting protein binding site, we are actually targeting histones. So these ones we are also bringing in proteins that can bind to particular histones at the regions you are interested in. So the regions you are not interested in will not have the proteins on them. That means they will be degraded or denatured, but not the ones with our histone modifying proteins. Okay, then you also work with them because that's what they are interested in. With the DNA with the DNA-seq, it's something similar to the first one, but in this case you're actually going in with a DNA, something that degrades the unwanted way, the unwanted part. So the DNA goes into cut like regions around where you want, so that you can isolate that particular part. Okay, so we actually look into them. So let me just take them one by one. So the first one, the DNA binding ChIP-seq, right? What does it detect actually? So it actually detects like, so let's the DNA bind the protein that we are this green protein itself is actually a specific DNA binding protein. So based on your gene of interest, you design a protein that can bind to your gene of interest, right? So it's more of a transcription factor. Like an example can be a transcription factor. So you design this protein to bind to your gene of interest, and it will go in, and mostly they bind to achromatic regions, because transcription factors actually they can't access heterochromatic regions, right? So if you are using transcription factor in this case, they mostly will detect achromatic regions but not heterochromatic regions. So the steps are like, first you have to stop the cell cycle. You have to arrest cell cycle. So in this case you can arrest cell cycle by using formaldehyde, right? Then you freeze like the entire thing, like the entire process, by introducing your protein. Then the DNA binding protein, which let's say we are using a transcription factor in this case, will go in and pair or bind to the places that it can recognize. Okay, so after a while, or after it binds, then we subject we subject the entire DNA to fragmentation, probably by sonication or using enzymes. Okay, so when we get a fragment, then we add, so in this case we are using antibodies. Okay, the procedure is called immunoprecipitation. So we use antibodies that are specific to the particular protein we used. Okay, so the antibody will just go in to pull the protein or release the protein. Then we can use our gene of interest. You see, we had like a long DNA, and our protein went in to pull or bind to a particular one we are interested in. So this is what we are interested in. So we just take our protein off, then work with this one. Okay, so whatever you want to do with the DNA, whether it's if you think the DNA is not enough, you can amplify using problem PCR and all those kind of things. Okay, so the second method is what is it? Histone modification ChIP-seq. Okay, so that one is actually similar to the first one, but as I said, we are not targeting the gene of interest. We are actually targeting histone modifications. So we are sending in a particular protein that can bind to the histones. based on their modifications. And I'm sure you know like the various histone modifications we have. I'm not sure if we've discussed them, but you should know at least from class, like the various lysine residues that are methylated or acetylated. Okay, and they all will determine whether that particular chromatin will be heterochromatin or isochromatin. Okay, so that this one to that particular protein you are using will go in to bind to your gene of interest. Similar procedure to this, so I won't waste time on this. Let's talk about the DNA-seq. Okay, so this one is actually not a technique that we use to look for proteins. Okay, so this one we mostly use it for looking for DNA accessibility, like achromatic regions. Okay, so in this case we send in a DNase, and I'm sure you know what a DNase is. An enzyme that cuts DNA, right? So mostly this DNase 1, we use DNase 1 preferably, right? Preferably we use DNase 1, and this DNase 1 is actually, it cuts preferentially in open regions, open chromatin regions. Okay, it also cuts in regulatory regions as well. It also cuts at enhancers and promoters, and you can just trace back the latter ones I said to achromatins. So promoters, enhancers, and regulatory regions, they all actually play a part positively in achromatic regions. Okay, so these are the places where DNase 1 cuts. So they actually they can't access close chromatin like heterochromatin regions. Okay, so you go in, you treat your DNA with DNase 1, it's going to cut the accessible DNA regions, or the regions that are achromatic, right? And it will isolate the short DNA fragments, like in this case. Okay, then we just add linkers and sequence the fragments we have. Okay, we do sequencing to know like the order of nucleotide sequence, blah blah blah, right? The last one is it stands for formaldehyde assisted isolation of regulatory elements. Yes. Okay, this one is actually a more clever method. So it uses a protein bound DNA cross-link. Okay, similar to this particular one, the DNA binding protein. So what it does is it does not, how should I put it? You let me not lie. Let's look at the steps one by one. So first, what you have to do is cross-link your DNA, the region where you think contains your gene of interest. You cross-link with the protein that you are bringing in. Okay, you fragment the DNA, the entire DNA, and just like in this first one, it's going to bind to only your gene of interest. So afterwards, you perform phenol and chloroform extraction. Okay, so after doing this, open chromatin regions are going to be separated. Okay, only the open regions, that means heterochromatic regions, are not going to be separated. So these are 4 methods that we use to ascertain whether a particular region of the genome is heterochromatic or heterochromatic. So there are some techniques over here. Okay, and each one of them does different things. So let's look at when we want to, I think this one is actually similar to what we discussed, assessing chromatin accessibilities. Okay, so assays that we can use to assess the accessibility of chromatin. So I think we can even use all these 4 as assay for chromatin accessibility. Okay, but the other ones like ATAC-seq. ATAC-seq stands for assay for transposase accessible chromatin using sequencing. Okay, and this one uses a transposase called TN5 to insert sequencing adapters into accessible chromatin regions. It is fast and requires low cell input. So this one will be like it inserts a particular protein. Okay, the transposase is a protein, an enzyme protein. Okay, it will insert it into sequencing adapters like the accessible regions. So in this case, wherever your transposase is inserted, it will be like an accessible region. That means like an chromatic region. The words are not coming. Okay, so this can be used to determine whether the region of the genome is achromatic or heterochromatic. Sorry. Another one is DNA-seq. This is like the second one on the diagram, the third one. Okay, we've talked about that. Fer2, we've talked about that. So another way is epigenetic profiling. So what assays do we use to look for, or what assays do we use to do epigenetic profiling? Okay, to check like epigenetic status of a particular person, right? So we have chip chromatin immunoprecipitation. Okay, we use this like, if you hear immunobia, that means it contains or it employs antibodies. Okay, so we use antibodies to detect histone modifications. So histone modifications are not genetic related, that's why they fall under epigenetics. If I say not genetic related, I mean like it does not affect like the original DNA sequence. Okay, so we use antibodies like through this method, chip. We use antibodies to detect histone modifications, okay, associated with chromatin, such as K9, the acetylation of K9 or methylation of K4. So mostly, if you see this kind of modifications, they are mostly found in chromatins. Okay, K9 being acetylated and K4 being methylated or acetylated. So mostly K9, K4, K14. If you see as like acetylation on these ones, they are mostly related to chromatin, okay, or making a particular chromatin more accessible. But if you see K9, K27, and those ones, methylation of those are mostly causing a particular chromatin to become more heterochromatic. Okay. Another method is N-chip, called engineered DNA binding molecule mediated chromatin. So the name is N-chip. N-chip uses molecules like CRISPR-Cas9 to capture. So these two, ChIP-seq and chip are ChIP-seq and N-chip. We use them to do epigenetic profiling. That there are also assays for transcriptome analysis. Those are RNA-seq or RNA sequencing and total RNA sequencing, or even you can do like specifically for mRNA sequencing. Okay, so the RNA sequencing will identify highly expressed genes. Okay, for that one, they are actually sorry, they are just sequencing the only the RNAs of your destiny. Okay, and you are trying to see which are located in achromatic regions. Of course, if they are in heterochromatic regions, they are not going to be expressed. So if you are looking for highly expressed genes, you are going to look for them in the heterochromatic regions. Total RNA or mRNA sequencing, that's something different, not so different, but it helps map transcript to the genome to identify the transcriptional hotspots. So after probably identify where the highly expressed genes are, we just use it to map the entire genome to see where transcription actually occurs like more. Okay, those are the transcriptional hotspots. There are also assays using cytogenetic and imaging techniques. So these are more imaging, like visual stuff, right? We have banding, R banding. I think reverse banding is like a reverse of J banding. In J banding, we use gemcitabine, right? So it does like the reverse of J banding. So let's read it. It produces a pattern that's reverse of J banding. So in J banding, in R banding, the active early replicating achromatic regions appear light and the heterochromatin appears dark. But in J banding, it would have been the achromatins appearing dark and the heterochromatins appearing light. Okay. There will be a diagram explaining this, so I wouldn't really talk much about the R banding and J banding. Another one for the cytogenetic and imaging techniques is DNA fish. The fish is fluorescence in situ hybridization. This is very common in molecular genetics, right? Fish, the use of probes to detect like gene of interest. Okay, so we use fluorescent probes. You design a probe specific to your gene of interest and we use it to directly visualize specific genome loci in the nucleus. So the idea is whenever that particular probe binds to your gene of interest, it gives off a particular signal. I don't know, mostly visually. Hey, why did I say I don't know? I know, because it is fluorescent. It's going to fluoresce, right, whenever it binds to your gene of interest. And another one is super resolution microscopy. This is like microscopy. Another so let's look at assays or techniques we use in sequence-based and computational methods. We do methylation analysis. Okay, there are so many of them. Example is bisulfite sequencing. And in methylation analysis, we use that one to identify CpG islands with low methylation. CpG islands are just like regions in the diagram, in the DNA nucleotide where you find like C gene, like C cytosine and gene guanine, like you find them right in a particular region. So let's say C gene hotspot, and the P is just like you see nucleotides are linked by a phosphate, so C phosphates, and mostly in these regions are where we find most of our methylations in our gene. Okay, so we use methylation analysis. When we are doing methylation analysis, we are trying to identify CpG islands with low methylation. Okay, and CpG islands with low methylation typically tells, or yes, it just tells us that that particular place is an active region or an achromatic region, right? So we mostly found them in active promoters. We also do GC content mapping. GC content is the achromatic regions are often enriched in GC content compared to heterochromatin regions. This thing. Let me see if I can find a way around this to explain. GC are more GC pairs are more stable than AT pairs. So if I'm going to talk about stability, probably we should find more AT in achromatic regions. But hey, they are saying achromatic regions are often enriched in GC content than heterochromatic regions. So we can use this particular GC content mapping to also tell if a particular region is achromatic or heterochromatic. The reason being is if you go to an achromatic region, you are going to see more EC pairs than AT. If you go to heterochromatic, you are going to see more AT pairs than GC, right? Sorry. Another method is proximity ligation methods. Okay, and this one maps 3D genomic organization to distinguish between A compartment, like active ones, and B compartment. Okay, this is not really important. There's a last one, which is DNA methylation mapping. So this one we actually use restriction enzymes. We call them methylation sensitive restriction enzymes. Okay, so these restriction enzymes, we use them to distinguish between the methylated part of the genome from unmethylated parts. And you know, highly methylated parts are mostly heterochromatin, and the unmethylated parts are equochromatin. So let's take an example from this particular diagram. Let's use this first one, the DNA binding chip-seq thing. Okay, this is a more detailed diagram. So let's say you have your entire genome, right? And let's say the red parts are where we are interested in, but if you get a DNA right now, you won't see a red part and you won't see a gray part. So let's say we are interested in a particular region, but we don't know where it is. So let's say we have two of them. Okay, the two genes, and we just design a protein that can bind to those regions. So these are our protein of interest, and our protein of interest will go into bind to wherever our gene of interest are. Okay, so after binding, we just subject them to fragmentation, and after fragmentation, you realize those without the bound, we are not even focusing on these ones. We are just focused on getting our proteins back, and in getting our proteins back, you are going to get our gene of interest bound to those proteins, right? Then we just use antibodies to pull off our proteins, and our gene of interest are going to release. Then we just prepare sequencing library and do whatever we want to do with it, right? Same thing for this as well. I think this is the histomodification stuff, but same thing. What is this? So let's look at metaphase chromosomes, right? So this is like we are entering the cell cycle now. So you see, we know, yes, I'm assuming you know what the M phase is, right? So as the cell enters the M phase, the level of compaction of the chromosome changes dramatically, and I'm sure we've discussed this, right? So by the end of prophase, the sister chromatids simply means like the two chromatids that come together to form the chromosome. You see, the chromosomes are something like this. So individually, something like this, and something like this. So like this green one is one sister chromatid, this another sister chromatid, right? So the two sister chromatids, by the end of prophase, they will be very like compacted, like entirely heterochromatic, and when they come together they will form the overall diameter will be 1004, because a highly compacted chromosome or chromatid is 700 nanometers. That's like the heterochromatic form, but the achromatic form will be like I think 300, right? These highly condensed metaphase chromosomes undergo little transcription, and it's of no surprise because they are what heterochromatic and they are highly condensed. In metaphase chromosomes, the radial loops are anchored to a scaffold, okay, and the scaffold is formed from the nuclear matrix, so they are actually like attached, or the scaffold is like just like their skeleton, okay. So we need, I think this point is actually established already. We need histones for that level of compaction of radial loops. Let's see what we have. So there are two multiple protein complexes, okay, that help organize metaphase chromosomes, and these proteins are condensing and cohesing. So from their names you can easily tell their functions. Condensing means they help condense the chromosome. Cohesing is like bring cohesing, like bringing them into cohesion, okay. So I'll actually get into that. So the condensing plays a role in compacting the chromosome, okay, like from the 300 nanometers to the 700 nanometers, and the cohesion plays a role in bringing the two sister chromatids together to form like the chromosome, okay. So these two proteins, the condensing and the cohesing, they both contain a particular like a category of proteins called the SMC proteins, and SMC is an acronym for structural maintenance of chromosomes, right? And these SMC proteins are ATP dependent. So let's look at this. You see, before the cell enters M phase, right, it is achromatin, like, or achromatic, right? Even though it is condensed, but it's not highly condensed. The diameter is 300 nanometers, so the radial loops are 300 nanometers. But as it exits the S phase and enters G2 phase and preparing to enter the M phase, you see in the interface, the interface, like the G1, S and G2 phases, there's condensins are mostly in the cytoplasm, right? These blue proteins, the cytoplasm. So after the S phase and the G2 phase, where the cell is preparing to enter the M phase, the condensins will move into the nucleus and help condense the chromatin, okay, and condense them further from 300 to 700 heterochromatins nanometers, right? And this will make it like more compact. So after, let's look at this diagram. So here they are. So initially, two sister chromatids are, you see, the cohesin is like the protein that brings the two sister chromatids into cohesion. So these red proteins are the cohesins bringing the two sister chromatids together, right? So this is before it enters the M phase. So at the beginning of the prophase, like before, like when it is exiting the S phase and about to enter the M phase, the chromatid or chromosome is now compact, right? It is now heterochromatin. So now we've entered M phase, and what happens in the mitosis? Mitosis, the cell starts to divide. That means each daughter cell will have like one of the sister chromatids. So we have to get rid of whatever is holding the two sister chromatids together, and those are the cohesins. So we destroy the cohesins, but those are the centromeres are like more stable, so that will be removed last. So that will be like at the anaphase, okay, and the last cohesins are the centromeric regions are going to be degraded. Then the two daughter cells will take so let's look at mitosis and meiosis, okay. In I'm going to take mitosis first. In mitosis, you see there are these sister chromatids. Okay, there are 4 of them. So individually, there will be like 8 chromatids, but the pairs there are 4 pairs. So they will line up in this is in prophase. In metaphase, they are going to line up in the center, like bind to spindle fibers and stuff. Then the cells start dividing, cytokinesis. Then each one of the daughter cells will get half of the chromatids, right? So this one will get 4 from the 8. That's mitosis. Okay, but for meiosis, the 8 chromatids, or the 4 sister chromatids, will pair up to form tetrads, 2 tetrads. In the first division, the 2 daughter cells are going to get 2 pairs of sister chromatids. Okay, so this step is like this step, just that they have half the number of sister chromatids. Okay, and this one also reduce. This one also replicate. I've forgotten the word. This one also replicate again, and the daughter cells will now have like two chromatids. Okay, so there's like meiotic daughter cells, and these are my mitotic daughter cells. You can see the number of, so this one will have like n number of chromatids, and this will have, in mitosis, the daughter cells will be 2n, right? I've said everything. So let's look at this one too. I've mentioned it. This is just telling us about the forms of folding. Was the folding of the chromosome, the chromatin? Okay, so from the 2 nanometer parts, that's at the interphase stage, J1, S1, S2 phases, right? The DNA will still be at the 2 nanometer region. Then the chromatin will come in, the histones and stuff, convert it into the 10 nanometer or the 11 nanometer structure, right? Then as it enters the metaphase, that's mitosis, it will go on to condense to 30, then 300 for chromatins stay so, but for the heterochromatic region, they are going to condense further to 700 nanometers. And I'm sure you are very well versed in cell cycle, right? I'm sure you know this. So let's look at the morphology of chromosomes. Okay, so chromosomes, should I say they have 2 parts? They are made in, they have 2 parts, like top and down, or upper and bottom parts. So these are chromosomes. Like the tips have the telomeric regions, and the center part is the centromere. Okay, and the 2 parts are the arms. The arms shouldn't always, or the arms are not always equal. So you can have a short arm and a long arm. Okay, the shorter arm is called a P arm, and the long arm is the Q. I don't know how you're going to remember this, but if the arms are equal, we call the chromosome metacentric, right? And if the upper arm is it's I don't know how to put this. If the lower arm is bigger than the upper arm, but not like entirely bigger, you see the difference between these 2 things. I don't know how to say them in words, but this is in both cases, the lower arm is bigger than the upper arm, but this one is bigger, the head of this one is bigger than this one. So if the head is a bit bigger than the other one, it is called submetacentric. So it's like a sub of this, submetacentric. And the one with the smallest head is called acrocentric chromosome, right? So, but this is very important, the P and the Q. Okay, so mostly a chromosome can be given to you without the name of the chromosome. You'll be given like a number like this, 17q11.2, and the lecturer or that particular person will expect you to know the chromosome and the position, the position on the chromosome you are talking about. Okay, so let's look at this. 17q11.2. Let's look at it. 17 means that chromosome is chromosome 17. Okay, Q means the type of arm, so we are looking for a place on the chromosome. The Q means whatever region we are looking on the chromosome is found on the Q arm. That means you're looking at this entire part. Then we come to 1. The first one will be the metacent, like there are stages, like the region, band, and subband. So the first number after the arm will be the region. So it will be, they are divided into 2 regions. one, like region 1 and region 2, right? So the 1 means it is found in the upper region, that's region 1. Then we come to the second one, we have bands. The bands, as we have 2 bands within each region, so that means on the arm we will have 4, on the lower arm, on the kill arm we will have 4 bands, right? But in the P arm we will have 3 bands, 2 1 1. You see, we have 3 bands. But in the kill arm we have 4 bands. So 1 means we are the top upper top, right? Then the point 2, now the sub band. The sub bands we have 3 3 3 3 3 1 2 3 4 12. So there are 12 of them. These are points 2, so obviously it's going to be here. So it's up to you to know how to read this if we give something like this. Okay, let's look at visualizing metaphase chromosomes. We have three techniques in visualizing metaphase chromosomes: phytohemagglutinin PHE, or using cosimid, demicolchin, or colchicine. I don't know if I'm getting the pronunciations right, but Charlie, that's my intuition. And also 3:1 methanol and acetic acid. So let's do some reading. Patient cells are incubated and undergo division. Blah, blah, blah. So when we are using the phytohemagglutinin, we use it to stimulate cell division. Okay, so these are just ways of playing around with the cell, depending on what you want to do with it, whether you want to arrest the cell cycle to do a particular test, or you want to induce or stimulate cell division to do something. Like, it depends on whatever your aim is. Okay, so if you want to stimulate cell division, you use the PHA, phytohemagglutinin. If you want to arrest the cell cycle, especially in the metaphase phase or metaphase stage, you use cosimid, demicolchin, demicolcine, and then colchicine. Okay, these are for arresting the cells in the metaphase stage. Okay, and the methanol acetic acetate solution is used to fix the chromosomes for staining. So not any chromosome at all, the metaphase chromosomes. So after using the cosimid and its friends to arrest the cells in the metaphase, if you want to fix them on slice, like microscopic slice for analysis, you use methanolic 3:1 methanol acetic acid solution mixture. Then to fix them. Yeah, let's go into the next. So this is where we are going to look at the J banding and the R banding, like, very well. Okay, so the visualization, like, after using the methanol acetic acid to fix your chromosomes for staining, the first type, the most common type of staining we do is the J banding, called the GEMSA banding. Okay, and that one we stain using GEMSA, and actually it was developed by someone called Gustav GEMSA. Okay, so in J banding, we will have light and dark bands. Okay, the dark bands will contain the AT rich regions, and sorry, as I said, I think, not me, as the slide said, the AT rich regions are mostly heterochromatic regions, and the GC rich regions are mostly echromatin regions, right? So in the GEMSA staining, the dark bands will contain AT, or the dark bands will be heterochromatins, and the light bands will be GC rich or echromatin. We also have KO banding, C banding, and R banding. R banding is the reverse of this. I'll talk about that. KO banding is, we use something called quinacrine fluorescent stain, and that's yellow. Nothing much was said about it. And C banding is only used in staining the centromere. Okay, this is not really important. It's only important when we are interested in looking at the centromere. The R banding, it does the reverse of GEMSA stain. We use GEMSA, right? We use GEMSA, but in our staining, the AT rich regions will be the white ones, and the dark regions will be the GC rich, right? So before we apply the GEMSA stain, we heat the chromosomes. Okay, before we stain, we apply the GEMSA stain. Okay, the reason why we do that is the AT rich regions are more readily denatured by heat. Okay, so I think the aim will be like denaturing the heterochromatic regions, which are AT rich, before we apply the GEMSA stain. So let's look at karyotyping. Karyotyping, I'm sure you guys have heard of it. We just use this to capture like a genome-wide snapshot of a particular individual's chromosome. So we will say this diagram, something like this, just to make sure you have we have a visual of like your set of chromosomes. Okay, the set of chromosomes of an individual. So I'm sure you also know this. A normal female has 44 pairs of 44 chromosomes. That's like 23 pairs, right? 46, and the last set is XS. Normal male 46, the last set is XY. Let's talk about evolution of human chromosomes. This one talks about some nasty shit. So we are going to actually base our discussion on these diagrams. Okay, so this slide actually says these were like chromosomes from primate ancestors. I don't know if these should be monkeys or what, but they said our ancestors used to have these chromosomes. Okay, and these are human beings chromosomes right now, and they've realized we actually contain the same chromosomes as them, or whoever these ancestors were. We have the same chromosome, just that there's been some shuffling around. So let's see some of these shufflings, the translocations. The major events, they call this evolution. They, not me. The formation of chromosome 2 in humans. Our chromosome 2 is formed by chromosome 9 and 11 of our ancestors. So 9 and 11, the red and wine, right? I don't know, me, I call this color wine. So the red and wine of our ancestors came together to form our chromosome 2. And also our chromosome 14 and 15 were formed by the breakage of ancestral chromosome 5. This yellow or orange, chromosome 14 and 15. So these are them. Okay, you can just take time and read through. I'll be wasting time on this. Doesn't make sense. Hey, sorry, I'm so this is a normal female, or like a normal female, right? You see it will have like 23 pairs of chromosomes. Okay, and this will be an aneuploid. Aneuploidy means like abnormal number of chromosomes. Okay, so in this case we are looking at 48 chromosomes. Okay, that means that particular female in question has attained 2 extra chromosomes, or an extra pair of chromosomes, XX. Okay, so it now has XXXX. Okay, so this is called aneuploidy. Aneuploidy can be gain of extra set of chromosomes or the loss of a set of chromosomes. We also have a condition called trisomy 21, and we are entreated to read on Down syndrome. This is the same as trisomy 21. So trisomy 21 simply means on chromosome 21, when it's 21, this, right, we've gotten like an additional chromosome, not a pair, like a single chromosome, making it 3 in the region. That's why we call it trisomy. These are abnormalities. We have translocation, like different parts breaking and substituting themselves. We have deletion, we have insertion. That will mostly be like translocation, or from a different place to insert itself, just like this. We have isochromosome. So instead of the sister chromatids separating this way, they will separate this way, like top and down. Instead of them separating left and right, they will separate up and down. This is called an isochromosome. They can form rings. These are all abnormalities. Okay, so let me drink some water. Oh, all right, so let's look at fish. Fish is used in detecting gross like chromosomal abnormalities. Okay, and I'm sure you know what fish is, like that fluorescent labeled probes thing, right? We'll look at them. So let's say we actually okay, sorry, let's say we are sensing a particular I don't want to call it mutation. Let's say we think we've seen we are detecting a chromosomal abnormality somewhere in a genome, right? And we have to detect it. So we just design a probe that's probably complementary to that region, okay, and that probe will be labeled. So in the fish case, it will be fluorescently labeled, okay, and the probe will go in to bind to where we think that normally has a case. Then we will use any visualizing technique we have to, you know. So so we have, in the case of chromosome, there's something we call multicolor fish chromosome painting. So in this case, you have like different dyes. So let's say when that particular fish binds to chromosome 1, it will be this color, chromosome 2 with this color, so that we can differentiate between like you can tell where that particular anomaly is, whether it's on chromosome this or chromosome 5 or chromosome 2, right? So each color will be a depiction of a different chromosome, okay. So let's look at different types of fish probes that we have. We have telomeric, or we have tel probes, okay, and as the name suggests, they are used to hybridize telomeric regions, and these are used to detect deletions or any anomalies at the telomeric regions, okay. So the tel probes, fish tel probes, are used to detect anomalies, mostly deletions and rearrangements at the telomeric regions of a chromosome. We have the sep, this like centromere probes. These are used to also, just like the telomere, this is more specific to the centromere region, to detect aneuploidy, okay, mostly during at the interphase and the metaphase stages. We have whole chromosome probes. So this is not like specific, it binds to the entire chromosome, and upon binding, just like these colors, upon binding to give a particular color. So this is used to characterize chromosomal structural changes. So if we are suspecting a change in structure of a particular chromosome, we use WCP to, study or characterize the structure of the chromosome, if truly the structure has been altered or not. We also have LSIs, and what are the names? I've forgotten their names. It's something identifier, locus specific identifier, right? So those ones are more like specific. So based on what you are interested in, we design that probe to bind to our loci or locus. The loci are plenty. Our locus, right? Yes, locus. Our locus. So we, upon binding, we use that probe to detect whether there's been any form of rearrangement, deletions, or amplifications. Okay. So these are the types of fish probes we have: tel, sep, WCP, and LSI. Okay. So let's look at genetic abnormalities by interface fish LSI probes. So greater or less than 2 signals per nucleus is considered abnormal. So the LSI, actually, fish LSI probe is a fusion type of probe. So in this case, like, the fusion simply means we are using two probes. Okay, one, mostly it is one green and one red, but the diagram we'll be using is green and black. Okay, so then don't get confused. So we are expecting like two signals. So if the signal, after using the probe, if the signal we use is more than 2 or less than 2, then obviously there is an abnormality or an anomaly. Okay, so if it is 3, mostly it's a trisomy. If it is 1, monosomy or deletion, right? So let's look at this one. This diagram actually is a bit confusing, but I'll try my best to explain it. So let's look at the first one. This diagram over the first one. So that's like in a normal person. Okay, so the first thing is, because we are using two type of probes, we expect that each probe is going to bind to, and in this case, one is black, one probe is black, another is green. Okay, so each chromosome should be colored green and black, like separately. So if a chromosome comes with both black and green, or there's translocation somewhere, okay, that means there's an anomaly somewhere. Okay, so we are actually looking out for like same color chromosome. Okay, so let's look at it. What's happening in gene A. For the first one, like this one. Okay, the gray ones, this is very normal, so I don't really want to speak about this, like the normal cell. So like when the probes, you see the probes are like specific to a particular chromosome, so it binds to the chromosome it was made for. It goes in to bind to it, no problem. It shows black color. It binds into this. It binds with this. It shows green color. So this is very normal. Let's talk about these two guys. The second one is your friend is saying. in the nucleus you see overlapping signals. Okay, so mostly we are looking at the color. So instead of getting two greens and two blacks, in this case you are getting like green, black, and an overlapping one. And mostly in this case, it's this is what it depicts. Okay, there's been a translocation somewhere. Okay, so there will be one fusion signal, one normal green, one normal black. That means number two they are messing up. Okay, and it mostly indicates translocation, or indicate that one chromosome carries a particular fusion, like a translocation from another chromosome. Okay, and mostly this type of translocations, they are found in chronic myeloid leukemia. Okay, let's look at the third one, which one it is actually similar to the first one but not like the same thing. So in this case, like both chromosomes, they have derivative. The derivative is a type of chromosome abnormality. I didn't mention it. Where is it? This one is a type of translocation or insertion. Okay, so it's a type of anomaly or chromosome abnormality. Where is it? So in this case, in the last one, both chromosomes are showing derivative. They are showing like the Charlie. They are all like not abnormal, right? So now what we see is two fusion probes, like the normal green, the normal black. Okay, and these ones are not very clear. We're going to see like a reciprocal translocation product. Okay, and a normal translocated chromosome, which is this one, like when two parts are rearranged. Okay, so reciprocal translocation is actually like two chromosomes that have exchanged segments. It's like translocation, but simply by changing a segment, you can call it reciprocal. Okay, so just like this chromosome and this so this and this, you can call this this green part belongs to this one and the black one belongs to this one. You can call reciprocal translocation. Okay, so this is not really important, but the focus is just how these fusion probes are used to detect anomalies. Okay, so mostly when you are getting two colors, you see the probes should bind, because we have two sister chromatids. You should have like two colors, like two I don't know how to put this. The same chromosome should give the same color. So for a particular color, you should have two probes. Okay, so for one chromosome, you should have two probes showing up for that particular chromosome. So for, let's say we have chromosome 1 and chromosome 2. For chromosome 1, you should have two blacks. For chromosome 2, you should have two greens. If you are getting more or less, that means there is a problem somewhere. Okay, that's what we use the fish LSI probes for. What is this? This is the whole chromosome painting. So in this case, we use, as I said, we use them for chromosome structural characterization. Okay, so probably after staining, you realize if there is any structural anomalies, you are going to detect that as well. So let's look at some uses of fluorescent in situ hybridization, which is FISH. So we use them in identification and characterization of numerical and structural chromosome anomalies. So we can use numerical anomalies is looking at whether you've lost some or you've gained some, like the aneuploidies, trisomy or monosomy. Okay, and the structural one will be using WCP. Okay, the detection of microscopically invisible deletions. We also use FISH for detecting deletions that we wouldn't be able to detect when using microscopy. Okay, and also detection of subtelomeric aberrations, and this is done by the help of using the TEL FISH probes. Okay, and also prenatal diagnosis of common aneuploidies. I think almost everyone can do that. So we use them in identifying and characterizing numerical and structural chromosome abnormalities, detecting microscopically invisible deletions, detecting subtelomeric aberrations, and prenatal diagnosis of common aneuploidies. So let's look at some this slide is saying truncations and translocations are associated with certain genetic disorders and blah blah blah. So as I said, the chronic myelogenous leukemia, the chronic myeloid, it can also be caused by. So let's define it here. I think I've mentioned in the previous slide. The CML, chronic myelogenous or chronic myeloid leukemia, are leukemic cells that contain shortened chromosome 22, okay, and a longer chromosome 9. So the chromosome 22 of a particular person is shortened and the chromosome 9 has been extended. So in this case, patients suffering from chronic myeloid or myelogenous leukemia have a shortened chromosome 22 and an extended chromosome 9, resulting from a translocation event in the kill arms, that's the lower arms of these chromosomes. So it's like the chromosome 22 will be broken and added onto the chromosome 9, right? So the shortened chromosome, which is the 22, is distinctive and called Philadelphia chromosome. Philadelphia. So we can use multicolor fish, right? Identifying this type of chromosomes, okay, like checking these Philadelphia chromosomes, multicolor, like if it binds, if a particular region of the chromosome is made up of different type of different part of different chromosomes, it will show different more than one color. So if you are seeing more than one color on the same chromosome, that means there's Philadelphia something going on, right? I think this is the last page. So this is talking about chronic myelogenous leukemia. They are saying it is part of BCR, breakpoint cluster region. So it is a gene called BCR. BCR stands for breakpoint cluster region from chromosome 22. Okay, so that part of the chromosome 22 is going to be fused. So I think it simply means the part of the chromosome 22 that's broken off is called the BCR, okay, and it is joined to a part of chromosome 9 called the ABL. So mostly we say we refer to chronic myelogenous or chronic myeloid leukemia patients as BCR ABL fusion. Okay, that means the BCR will be broken off from chromosome 22 and joined to ABL of chromosome 9. This abnormal fusion will generate a protein called P20, P210, or sometimes P185, right? And the ABL of the chromosome 9 carries a domain that adds phosphate groups to tyrosine residues. Okay, so the fusion between BCR and ABL, the gene products we get from this fusion will also be a